Good afternoon, everyone. This is Suez from the Department of Bioinformatics, Sarda School of Engineering, Chennai. So today I'm going to present on the topic Machine Learning in Leukemia Treatment, QSI Modeling for Lead Compound Identification. So I know this topic sounds too complex for you guys. So let me break it down. Assume you're a doctor and a patient comes to you describing a set of symptoms. So you run some tests and do some diagnosis, and you finally come to the conclusion that the patient is suffering from leukemia stage 2. So you have two options. So either to choose for the existing drug in the market, or else you have like large database present in your web server, and you have to identify which will be effective for this disease. So that's where my project comes in on computational drug discovery, which is the machine learning in leukemia treatment. So the main agenda of my topic today is what is leukemia and what causes leukemia, and how to target the disease-causing protein, and what is computational drug discovery, and how does ML aid in computational drug discovery, especially leukemia. And I'll give you an overview of my web app which I've developed as part of my project at Celestra, and I'll also tell you how to evaluate the drug likeness of the compound that we have discovered. So what is leukemia basically? Basically, you probably know that leukemia stands nothing but blood cancer. So basically, this is due to large production of abnormal or immature leukocytes. Leukocytes are nothing but the white blood cells present in your body. So there are several types of leukemia. So among that, acute myeloid leukemia is the most prevalent form, and this is due to the abnormal differentiation and proliferation of HSC. HSC stands for hematopoietic stem cells. So because of that, there is overproduction of neoplastic clonal MSC, myeloid stem cells. Because of this tumorous myeloid stem cells, they produce this immature leukocytes, and which causes the progression of leukemia. So basically, if you look at the impact, we can see that males have like higher incidence and mortality rate compared to females, and you can check the statistics over here. So what basically causes leukemia? So the scientists have discovered that tyrosine kinase is a major protein that basically causes this disease. So there are several drugs in the market like midostaurin, glitinib, phenylenib, lilostorin, and sorafenib. So basically, I'll give you an example. So basically, consider this as like a bowl, and there's like an empty space inside, and that is called as your active site. So now you need to block this protein. So in order to block this protein, there are several drugs in the market. So why use of new compounds? So in order to like, there can be better compounds better than this, right? So that's why I'm using this computational drug discovery. Could there be other drugs that are effective than this? Probably yes. So like why this computational drug discovery is basically, so we have like a large number of compounds. We cannot just synthesize each and every compound and not just test each and everything, right? So it's basically becomes really expensive. So that's where my project comes in. So basically, computational drug discovery is nothing but the usage of ML algorithms and computation tools, and we identify the best compound, and we take that compound to the biotech people, and they prepare the drug, and we do for testing and training, and then finally release it to the market. So basically, the methodology which I've followed from my project is basically, so I've taken the data set from the chemical database, I've cut down the majority, like 6,283 compounds to 3,119 compounds to as much as 3,000 compounds. So basically, I've done all the filtration, labeling, and all. So I'll send you my PPT so that you can refer to that methodology which I've followed. So if you basically look at my web app, so basically, you can give your input molecule, whatever the database molecules are having, and you give it to my app, and it basically gives the predicted bioactivity, which is TIC50 value. So these are some of the bar graphs that I've generated using Matplotlib and Seaborn. So basically, you could see I've done some graphs. So you can see all the graphs, and I also compared the various machine learning algorithms. So I've used lazy predict classifier. So if you look at that, you can see all these graphs, and so what's the inference basically? So among all the machine learning algorithms, I found out that the random forest regressor came out to be better effective one, showing the accuracy of 85% for my project. So you can just directly log on to my website, drugsfortyrosinekinase.streamlit.app, so that if you're inventing some new novel drugs, so basically you log on to this website, you can give your compounds, so it will basically give you the predicted bioactivity. So this is like my website, as you can see. It's an app built using Python and streamed by me. So you can basically click on browse files, take your input molecule, and then you click on predict. It will start processing, and you'll get the predicted output. So here you can see, so the first molecule 6.2, 5.8, 6, 6.69, and 6.9. The fourth molecule, like the fifth molecule over here, is showing the highest predicted bioactivity. So I've used machine learning algorithm, so which is the random forest regressor, and you have taken that model, and then you have predicted the output. So So I can see Campbell 364623. So I've taken this ID, went on to PubChem website, and then I've noted down the name, which is the N2-chloro, which is the very big name. So I've taken this compound, I've downloaded the structure of it, and I've told you that targeting tyrosine kinase protein, right? So I've downloaded the structure of that protein. So basically combined together. So basically what like drug discovery majorly deals with is, so you have a protein, you have a compound, so basically dock together, like combine together, and you predict the binding affinity. So if the binding affinity is greater than -6, then it means it's like a better compound basically. So here is the compound N2-chloro. So you can see the hydrogen bonds. This is the protein, and this is my compound. So this is the lead compound, right? So if you like have any questions, you can just contact me through my email or Instagram. Thank you. It's by US.
US government. Will it be applicable for Indian data?
Yeah, basically there are several drugs that are approved by FDA, Food Drug and
This is
Administration.
data not government or in
There is no like Indian database available for this drug industry. We have
You
like
think
US.
you say that there is no blood cancer research is
We
being
have,
done in India?
we do have, but we don't have like the Indian website that has all the data stored. So basically, if you want to have any data, so basically we have like three databases. One is Germany one is Japan one, and the other is the US one. So basically, these three databases.
How many algorithms you have taken into picture and say that random forest is the best one? What are all the algorithms?
So
that-
basically, we built all the regression, linear regression, logistic regression.
Can you show that slide where all the algorithms were there?
So basically, you can see that the RMSE has been mean square error. So you can basically see the random forest has mean square error. And then if you look at it, the random forest regressor has highest R square value, and time taken is very less. So random. So when considering all parameters, I found out the random forest regressor has the best. And for this, how to like evaluate all, use lazy predict classifier module. So if you take that, you can basically compare all the machine learning algorithms.
So you have used a tool for this.
Like there is like a module present in Python.
What is the tool?
Lazy classifier.
Spell?
LAZY CLASSFI.
So you can take all the classifiers in one go.
Yeah, I can compare all the models.
How many records are there in the data set?
So I've done all the filtration data to get one. So it has come around 3,000 compounds.
3,000 compounds in the sense 3,000 attributes you are meaning or 3,000 records you are meaning?
3,000 compounds which are present in the database, which has all the things like hydrogen bonds, hydrogen oxide.
What are the attributes? How many attributes are here?
So here basically you can see the have. So I've taken molecular weight.
This one is box plot, right?
Yeah, oxygen. So number of hydrogen bonds.
How you are giving for each and every attribute you are giving a box plot?
Yeah, comparing the inactive and the active. So there are several compounds. So some drugs are inactive for leukemia and active for neurodegenerative disorder. So basically it's classified as for that. And how did I take this parameters? Basically there is like a established institute and like Pfizer, right?
Since it is a classification, right? You do not use it into regression models, right? So you are going to predict some compound's value, and based on that you are going to predict. So where is that content will be available?
So basically you have my average.
What protein? What compound did you go for prediction? You have taken some tyrosine, right?
Tyrosine kinase is my enzyme. So basically my model is tyrosine kinase. So we have compounds from the database. Whatever the data compounds you give to my app, so it will give the PIC 59%.
So that is the prediction value.
Yeah, that is
PIC.
predicted IC.
Okay.
Inhibition of concentration.
So do you have next thing for this output? How prediction you will be going for it? We have that in reports, right? We get some value, we get normal value and it's something like that. Do you have it for this type of values?
like you mean the reference values.
Yes, if it is normal or
There is
above
like
normal, we have to go.
So I'm actually writing a paper on this. So basically I've done all the paperwork with that. So basically there's a paper which states so the threshold value is 5. So if it's less than 5, it's inactive. 5 intermediate, above 5, okay.
Thank you, Vivek.
Thank you. Next,